So today's topic is cell signaling. We spent a lot of time talking about this. Go spend some more today trying to make sure you know how to do it. We talk about cell signaling in different ways. One way is local signaling, and that means that two cells that are just beside each other, two cells that are adjacent to each other, are communicating. Several ways that can happen. One way that we talk quite a bit about is these things called plasmodesmata. Where are those found? Plants. What complicates communication between plant cells? The wall. So this is like a tunnel between the walls that lets materials and messages go from cell to cell. What's the same kind of thing called in an animal cell? It's called a gap junction. So those are connections between adjacent cells that let them communicate. And what was a good example of some cells that was really important in animal cells? Your cells. The heart, right? Because what do the heart cells have to do? Yeah, they have to coordinate their beating, right? They have to all beat at the same time. Otherwise, you got some serious issues. All right, another local signaling thing that we talked about with the immune system is that sometimes the proteins on the surface of the cells, the MHC proteins, those proteins on the outside, they can bind together with those from another cell, and that can communicate. So let's go to a picture. This diagram we've seen a bunch of times. What's this purple cell? Not a bacteria, but just some kind of phagocyte, right? It's a cell that's eating things. What are those big cells, immune system cells that eat stuff? Close. Macrophage. So here they're eating the germ. Let's say that this thing right here is a virus. So what do we call the process when it pulls it in? If it's something big and solid? Phagocytosis. And it puts a little bubble around it, and we call that little bubble a what? Vacuole or a vesicle. And then what organelle is going to pull up beside that vesicle and dump stuff in there? Lysosome. It's going to dump in really strong enzymes, and it's going to carry out what process? There you go. Intracellular digestion. The thing's going to get broken into pieces, and these little orange triangles represent those pieces. What are we going to call those little triangles? Antigens. And the antigen is a word we're going to use for a foreign what? Protein. All right. Then this green thing, what's that called? MHC class 2. Only like phagocytic immune system cells have those. It's going to grab onto some of those little antigens, and it's going to display them on the surface of the cell. And we call that process what? Antigen presentation. And it's presenting those antigens to this cell. And what's that cell? It's a helper T cell. And over again, I told you here that the helper T cell, it's got little surface proteins that are binding to that antigen. And I said it's sort of going Helen Keller on it. And what I meant by that is it's touching it, it's connecting to it, and it's trying to get what? What is the top cell trying to transmit to the bottom cell? Shape. So it gets that shape. It activates this T cell. And we compare this T cell over and over again to what historical person? Paul Revere. Because it's going to send out its own signals. Signals that will either activate the antibody system in your body, the antibody-mediated system. What are the main cells involved in that system? The cells that make antibodies. The B cells, the B lymphocytes. Or it might activate the other part of your specific immune system, and what's that called? What's the part called that kills your own cells that are infected with something inside? Cell-mediated immunity. Regardless, this cell, this helper T cell, has to send out signals to all the specific parts of your immune system. That's pretty important. So that's one way these membrane proteins can help cells to communicate. We also got this cell over here, which is just a regular everyday cell. What's the word for a regular everyday cell? Somatic. And all your cells, all your somatic cells, have a little protein on them too. That's called an MHC what? It's 1. What do they display on their proteins? Proteins from inside themselves, which should be normal proteins, right? Unless what? Unless they're infected with something, or maybe they become cancerous. Either way, they put proteins out here. This is still a helper T cell. It binds to it. And if it finds a protein there that's weird, it's going to signal what kind of cells to come in and take care of this. Killer T cells, or sometimes they're called cytotoxic. I missed the TO there. You get it though. It's not cytotoxic, it's cytotoxic T cells. And those cells, we said, come in and they'll bind to this antigen, and then they'll sort of poke holes in the cell and kill it. But they're specific. Everything in this immune response you're looking at is specific. It only reacts to one particular way. Antigen, one specific protein. So this is just a good example of how these membrane-bound proteins let one cell send signals to other cells. And that's local signaling. Another example is what we got going on here. We've got two nerves connected together, 2 neurons. And what on this neuron, the first neuron, connects to what on the second neuron? What's the connection between what parts? Axon to dendrite, right? And what do we call that connection? The synapse. And here's a little more zoomed in version of it. So we've got an electrical signal coming down this neuron. What's the word for that electrical signal? Action potential. And when it gets near the end, there's an influx of what ion into this axon? Calcium, Ca plus 2, 2 plus, however you want to say it. And that forces out these little vesicles. It helps to push them out, causes exocytosis to happen. What's in those little vesicles? Neurotransmitters. And there's lots of different neurotransmitters. There's a couple you might want to just know if you hear the name. It's a neurotransmitter. Acetylcholine is one. Dopamine, serotonin, and there's lots of others. Those are just some examples of different neurotransmitters. Those neurotransmitters will then go across the gap from the dendrite. So this is, from the axon to the dendrite. So this is the beginning of the dendrite over here. And it's going to bind to these little receptors. What do we call those little receptors on the dendrites? What kind of receptors are they? Ligand-gated ion channels. And when the neurotransmitters bind to them, like always, they're proteins. What happens when something binds to a protein? Conformational change of shape. And when it changes shape, what happens to this thing? What is the change in shape? What happens to these receptors when they change shape? What do they do? They open, right? Because they're channels. They're ligand-gated ion channels. And what ion is going to start shooting into this postsynaptic neuron? Sodium. And that's the beginning of the next what? Action potential. Because moving ions are what? Electricity. That's how the electrical signal jumps. It doesn't really jump. It's not like a spark. But it moves from presynaptic to postsynaptic neuron through neurotransmitters. Another example of local signaling. Cells that are nearby communicating with each other. Make sense? Yeah.
Usually are those cells?
How long is like a neuron?
Yeah.
Most of them are really, really tiny, like microscopically tiny, but there are some neurons they're the longest. Some neurons are the longest neuron in your body. Like there's some that might be the length of your arm. Microscopically skinny, but crazy long. Think of it almost like a piece of wire, which is essentially what it is. But most of them are short. There are a few examples that are really, really long. All right, so let's go to long distance signaling. And probably the best example of this to think about are hormones. What are the parts of your body called that make hormones? What are the different things in your body as a group called that make hormones? Endocrine, right? Endocrine glands. And the way they travel is through what? Through the blood, right? They're released from the gland into the blood, which means they go to which cells? All of them. But are all cells affected by every hormone?
No.
Nope. Only ones with what? The right receptors. Because just like enzymes, receptors are specific, right? Only a certain thing will fit into the groove on it. What do we call cells that are affected by a particular hormone? Targets, right? They're target cells. So hormones travel from an endocrine gland through the circulatory system, through the blood, bind to receptors, and affect specific target cells. What structure in your brain controls the endocrine system? Hypothalamus, right? And one of the main purposes of the endocrine system is to keep you where? Keep you in homeostasis, right? It helps to regulate things like blood sugar, sodium levels in your blood, calcium levels in your blood, all kinds of stuff. It's about homeostasis. What gland is located just underneath the hypothalamus that makes stuff like growth hormone and ADH? Pituitary. What's the one that makes insulin and glucagon and helps to control blood sugar? Pancreas. And which is the one that makes epinephrine? Adrenal, right? Epinephrine is adrenaline. We'll talk more about those. We're going to have an endocrine day one day in here before too long. All right, pheromones, remember, are chemicals that are made and usually released maybe into the air, maybe in urine, maybe in sweat. They usually travel through the air, though. And what kind of signals are they mostly sending? Mating, attraction. It's about attracting mates, mostly. There's some exceptions to that rule, but mostly pheromones travel through the air and they help to attract mates. And they can travel not just from one part of the body to another, but for miles, right? Powerful, powerful things. Remember that plants also have some hormones. And most of those hormones in plants travel through the phloem. The phloem is the tube that usually carries what? Sugar. Sugar from where toward where? Yeah, source to sink. What is the source? Where's the sugar made? In the leaves, right? That's where photosynthesis happens. And the sinks are either the stems or further on down, the roots. There are a few hormones, and we talked a lot about this when we were dealing with hormones, in plants that can move through the air. They're gases. And the really famous one of those is a hormone called ethylene gas. Anybody remember what that causes to happen in plants? It causes their fruits to ripen. And we said normally it's not a huge deal because what happens to most of the ethylene gas that's released from an apple tree, for example? Just diffuses all out into the air. But when can you really make your fruits ripen fast? Put them in a bag, because that container holds in all that ethylene gas and it just keeps making it ripen faster and faster and faster and faster. Some plant hormones can also move from cell to cell, like between adjacent cells. And how do they get between those adjacent cells? The plasmodesmata, that's one of the things that it does. So I told you when we went through this the first time, this is the most important slide in the whole PowerPoint. The 3 stages of cell signaling. The last 2 or 3 tests, AP tests, there's been a free response question almost directly related to this slide in one way or another. Step 1 in cell signaling is reception. What's the word, generic word for a signal? Ligand. Right? So the ligand binds to a receptor. Most of the time that receptor's on the cell surface. We call those what kind of receptors if they're sticking out? Extracellular, right? Most of them are there. There's a few that are inside, and we call those intracellular. Why are most of them on the outside? What are most signals made out of? Proteins, which we know are not only big, but also what? Polar, charged. Those things can't get through the cell membrane. So most of the ligands have to be on the outside. What kinds of ligands have, or signals rather, have receptors inside? Well, most hormones are protein too. Steroid hormones, like estrogen and testosterone, because steroids aren't made out of protein, they're made out of what? Lipids. And lipids can go straight through other lipids, right? Because they're both what? Non-polar. They can go straight through without a problem. So step one happens. The ligand binds to the receptor, sort of like what binds to an enzyme. Substrate. They have to fit. The charge, the shape, it's all important. And just like what happens in an enzyme, you get a change in shape, conformational change or an induced fit. And in most things, that transmits the signal from outside the cell membrane to just inside it. And that triggers step 2, which is transduction. It's the complicated part. It's when the cell, I told you to think of 2 words when you hear transduction. What were those 2 words? Translating the message into something the cell can understand and amplifying the message, which means spreading it throughout the cell. Not from one cell to the other cell, but throughout the whole cell. Fast. Remember that this thing is usually really complicated. I compared it to what kind of machine? A Rube Goldberg machine, where you have all these crazy steps to do something really easy. And most of the steps involve transferring what kind of thing from one protein to another? Phosphate groups, right? What do the phosphate groups bring with them when they're transferred? Energy. So it's kind of like turning things on. And what do we call the enzymes that transfer those phosphate groups? Kinases, protein kinases. And so it's one protein turning on another, turning on another, turning on another. But it's not just a line. What happens to that line along the way? It splits, right? And all those splits are doing what? Every time the line splits, what's happening? It's amplifying, it's spreading that message out. But we call that series of crazy phosphate producing steps the signal transduction pathway. Or another term we might use is what? Phosphorylation cascade. And that keeps happening until eventually the thing that you're actually trying to turn on gets turned on. And then that causes the response, and that's the last step. So in the response, some kind of crucial cell activity is activated or maybe deactivated. It could be turning on or turning off a gene. It could be turning on a certain process in the cell, like a certain kind of chemical reaction, like breaking down glycogen. It could make a protein. Any kind of thing like that. But 3 steps. Reception, ligand binds to receptor, causes conformational change. That triggers transduction, which translates the message, spreads it out throughout the whole cell in a crazy set of phosphorylation steps. And then we get a response. Turn something on, turn something off. Turn on a gene, turn off a gene. Turn on a chemical reaction, turn off a chemical reaction. Let's talk about an example of this that we went through earlier. Find my picture that I want here. All right, one example that we talked a lot about and we watched a video about was epinephrine. Now remember, epinephrine is also called adrenaline. And let's say that this is epinephrine right here. And this is a liver cell. Cell in your liver. So epinephrine comes in and binds to a receptor. The kind of receptor that it binds to is called a G protein receptor. It's got to fit, just like a lock and a key, just like with an enzyme. And when it does, When it binds to that receptor, what happens to the receptor? Change of shape. And in this case, the change in shape that happens is that this little G protein that's normally attached up here, this lets go and slides down the inside of the membrane. And notice it's got some GTP with it. GTP is essentially the same thing as what? ATP. So it's taking energy from this G protein down here to this enzyme. This enzyme down the way, at least in the liver cells, is called adenylyl cyclase. When it gets phosphorylated by this GTP, it becomes active. And its job is to turn ATP into what? CAMP, cyclical AMP, which we refer to as what kind of thing? Secondary messenger. And this enzyme doesn't just make one of those CAMPs, it makes bunches of them. I'll just use these arrows to represent that. And those CAMPs move out through the cell, they spread the message, and then they go out, and each one of these activates what? A kinase, which activates another kinase, which activates another kinase, which activates another kinase. Until eventually in the liver cell, what's adrenaline telling that liver cell to do in the first place? Yeah. The liver cell is one of the main places in your body you store this thing, glycogen, which is an example of what kind of sugar? A polysaccharide, made of bunches of what monosaccharides attached together, glucoses. So this pathway is trying to tell enzymes to break down glycogen and therefore to put glucose into the blood. How would adrenaline tell the liver to do that? You need more energy, because what's adrenaline preparing your body for? Fight or flight, and you got to have energy to do either one of those. So that's a good example of how this whole pathway works, from ligand binding to reception, the transduction going on here in the middle, to the ultimate response being the release of glucose into the blood. Does that make sense? All right, we also talked about two other kinds of receptors. I'm not going to spend too much time on those. We just finished a minute ago talking about these guys, ligand-gated ion channels. And we said they're normally found where? At the synapse, on what structure in the synapse? It's dendrites. They're on the dendrites. And what kind of ligand binds to them? Neurotransmitters, right? The neurotransmitter is the ligand. And when those neurotransmitters bind, they cause that channel to open up. And when it opens up, what process can happen through it? What's the process called that moves something from high to low, but through a channel? Facilitated diffusion. It causes facilitated diffusion to happen. And in this case, it's mostly the facilitated diffusion of sodium ions. We also talked a little bit about this other kind of receptor. Not much. We didn't touch too much on this one. Receptor tyrosine kinase receptors. Scientists think that those G protein receptors were probably one of the original kinds of receptors in living things. But in the G protein, one of the disadvantages of it, each receptor can only activate what? One thing at a time. So is it going to cause a response that's like crazy fast? Nope. So we've evolved other receptors, this one in particular, tyrosine kinase, which has 3 phosphorylation sites on it. And it can, actually 6. And it can phosphorylate 6 things at one time. And it can cause a much faster, much bigger kind of response. That's not all I'm going to say about those for right now. Just realize that's a kind of receptor that can cause things to happen faster than the G protein. All right, so we talked about protein kinases a lot. We know they transfer phosphate groups from ATP to a protein, and that usually activates things. They're involved in all kinds of stuff in your body. About 2% of your DNA codes for protein kinases. That's a lot. There's also, though, the opposite of a protein kinase. We haven't mentioned those in a while. Those are called protein phosphatases. So they take away phosphates and therefore take away energy and usually turn things off. So on switch right here, off switch right here. Ways to activate and deactivate certain kinds of pathways in your body. So there we see a phosphorylation cascade. Ligand bonded to the receptor, activated a kinase, which then activated another one, which activated a different one, which activated a different one, ultimately caused the response. This picture is misleading though, because it makes it look how? It makes it look like a line. It makes it look like it's only going from 1 to 1 to 1 to 1. But really, it's going to look more like this. There's going to be multiple lines spreading the message, amplifying the message, spreading it all throughout the cell pretty fast. Let's see. All right, we talked about that already. Looking for something different. All right, remember that these ligands are specific. They have to bind to specific receptors, like an enzyme and a substrate. The same ligand, though, in different parts of your body can cause very different things to happen. Like epinephrine, for example. In your liver, it causes glucose breakdown. That's the message it's sending to your liver cells. But what message is it sending to your heart cells? Speed up, beat faster, right? What message is it sending to the cells in your around your pupil? Dilate. What about in your bronchial tubes? What do you want your bronchial tubes to do during fight or flight? Open, so you can breathe deeper and get more oxygen in there. So that same ligand can send a completely different message to different parts of your body, because the message is specific to a certain body part. The other thing that I want to mention, just to remind you, thought I had a slide on here, but I guess I don't. We said that steroids have receptors usually on the inside of a cell, intracellular receptors. And the two main steroids you need to think about are estrogen and testosterone. What's the advantage of having your receptors on the inside? Yeah, they can cause faster things to happen. Because if the receptor's on the outside, that message is first got to be transmitted from outside to inside. That takes several steps. And then it's got to be spread after that. Steroids can go straight through the membrane, bind to a receptor, and mostly what do they cause to happen? What do steroids cause to happen in your body? Once they bind to that receptor, they go straight to where? This is testosterone, and this is its receptor. They bind together, and then where do they go? They go into the nucleus, attach to the DNA, and cause transcription and translation of specific genes. That can happen fast. That's why these guys who are at the gyms and muscle heads, they get on the steroids, and in a matter of a couple of months, this little skinny dude is pumped. Steroids work fast. They cause fast responses. All right, does that make sense? All right, I'm going to take a break here for just a second. We moved a little faster than I thought we would there. Before we switch PowerPoints, I want to go back over this slide for a second because it's really important. Immune system slide. We've seen it probably 10 different times in class. But just a reminder of some things. So some germ gets in your body. You're exposed to an antigen for the first time. We call that what immune response? Primary immune response. What do we know about the speed of that response? It's kind of slow, 5 to 7 days or so. And what do we know about the amount of antibodies you make the first time around? Not so many, right? Slow, and it's not very big. But anyway, the way it happens, usually a macrophage will engulf that germ, do intracellular digestion on it. On its class 2 MHC, it'll present those antigens to the helper T. And then the helper T will send out its own messages, which we call what? What are those messages called that helper T send out? Interleukins. And that means between what? What's leuco? White blood cells. Leuco means white. Between white blood cells. And that stimulates B cells, but only the B cells that are what? Can make antibodies against that specific thing, right? Because every B cell can't make antibodies against everything. There's specific ones that can only make certain antibodies. So those get activated. And the first thing these interleukins are telling those B cells to do is to do what? There's only a few of these guys specific to that antigen, so what does it tell them to do? Replicate, clone, make many, many copies of yourself. So they do that, and then most of those B cells that clone become plasma cells. And they're called that because they have a whole lot of what organelle inside themselves. Rough ER. What do we know rough ER is about? Making proteins, shipping them out. Antibodies are proteins, right? So they make lots of proteins, thousands and thousands of them per second. Shoot them out. Antibodies that are specific to that particular antigen, the one we started with. Remember, though, that a few of those B cells never turn into plasma cells. They hang around as memory cells. They know how to make the antigen, right? But they don't. They're waiting, and how long are those going to stay around? Maybe for your whole life. How long are the plasma cells going to stay around? Just for a few days. And then they do what? What's the word? Apoptosis. Question.
So if like your primary antigen exposure, if the B cells are specific to antigens, how are they already there if they've never seen that antigen?
They're, let me think of the best way to say that. You only have a few genes to make antibodies, right? Handful. But what have we said about those few genes? How can a few genes make hundreds of thousands of different antibodies? What do we talk about when we talk about RNA processing? The different things can be treated as introns at different times. So these cells aren't making that particular antibody, but they're stimulated, and then they do the alternate RNA processing, and then they make the specific antibody. So it takes a while. You only have a few kinds of B cells. But those B cells can be stimulated to make a particular antibody. I didn't explain that very well, I know, but scientists are really still just trying to get to the bottom of how these few genes make all these antibodies and protect you from all these different things. That immune response, though, that's happening over here, it's called the antibody-mediated, or sometimes it's called humoral, because antibodies can only help you if the germ is where? Outside the cells. Humors mean what? Fluids, blood, fluids between your cells. The other part of that immune response is going on over here, and this is called cell-mediated immunity. And it's stimulated when a macrophage ends up, not necessarily a macrophage, when a helper T cell bumps into a somatic cell that's displaying some weird antigen, like a viral antigen. And then the helper T cell stimulates the killer T cells, the cytotoxic ones. They go out and they look for that particular antigen, that particular viral antigen, and any cell that's displaying that antigen, they essentially go Wolverine on it, poke holes in it, and kill it. So these two systems are both part of your specific immune response. This part kills your own cells that are sick so that they don't spread it. This part kills bacteria or viruses that are floating around in your body fluids. All the parts create memory cells. So you have memory T cells, you have memory helper T cells, you have memory B cells, so that the next time you get a disorder, let me go to a blank slide here for a second. We'll just use this. Let's make a little graph here. So let's say the first time you're exposed to a germ, your antibody levels start out at pretty much 0. But after you're exposed, it's kind of slow, but you get a response. And then what happens after you get well? Antibody levels drop down to almost 0. But what's still floating around in your blood? Memory cells. So that may stay like that for a long time. This is what we call what immune response? That's the primary. But let's say over here, right at this point, you're exposed to that same germ again. What happens? That kind of thing happens. And that's your secondary immune response. It happens way faster, it happens way bigger, because what's already there? Memory cells. And I think we're going to take the time to go into the endocrine stuff. So let's talk about one more part of your immune system. That was the specific part of your immune system, or sometimes it's called choir. Because you have to build that part up, right? You have to be exposed to things, you have to fight battles, you have to win battles, and then you become immune to things. But the other part of your immune system is called the innate, or non-specific part. What's innate mean? Born with it, built in, automatic. And by far, what's the most important part of that non-specific immune system? Skin, the wall, an active wall. Your stomach acid kills things that come through your mouth. What was the local flora? What's that? Flora. Those are the bacteria that live on and in you. And just because they're there, what does that mean? Other things can't be. It's like an offensive line. They're blocking for you. What's another part of your non-specific immune system? Well, yeah, other than your skin. Inflammation, when you get a burn or a cut or a scratch. Certain cells in your body send out this signal, histamine. And what does histamine tell the blood vessels near the damage to do? Open, expand, dilate. That sounds like a bad idea. But why do you want the blood vessels near an injury to widen? To get more platelets there to help start clotting it and fixing the damage, and to get more white blood cells there to kill the things that are coming in through the break in your wall, right? And again, there's more parts than this, but those are probably the main ones. Non-specific stuff, parts of your immune system that just protect you from general stuff, not anything in specific, just things in general. That is where we'll call it today. Thank you guys for coming today. I didn't expect hardly anybody to be here. You did good.